Good afternoon. First of all, thanks to the organizing committee for selecting our work as an oral presentation. My name is Paco Romero, and I am a Marie Curie postdoctoral researcher in the post-harvest physiology, pathology, and biotechnology group at the IATA in Spain. In this talk, I want to show you our last results about the loss of tomato fruit quality as consequence of a deficient copper availability. Copper is an essential micronutrient for all living beings, but it is also toxic when in excess. For humans, copper can only be acquired by the diet, and both a deficient or an excessive copper uptake have consequences in our health. Therefore, optimizing the copper content of the products of our diet is crucial for our wellness. As shown in the figure, mild or severe copper deficiency can lead to anemia, neurological, immunological disturbances, and among other symptoms. On the other hand, copper poisoning has metabolic consequences such as the Wilson and Addison disorders, as well as several multi-organic alterations, including muscular, liver, and kidney damages. Plants are also sensitive to this copper duality. In fact, copper deficiency growing conditions have been associated with low yield, developmental failure, and infertility of the crop. Also, low copper availability can be related to low nutritional value of the products. This problem has traditionally been solved by adding copper-based fertilizers to the soils, which recovers the yield, but has a high environmental cost in terms of soil and water pollution, and might also generate phytotoxicity when it is overused. But nowadays, the use of these agrochemicals is necessary, since about 20% of the European arable land is defined as copper deficient, and such percentage significantly increase depending on the region considered, as you can see here in the map by the blue and green colors. Therefore, agriculture is facing a challenge where the type of soil, the pH, or the organic matter content might make necessary the use of copper-based agrochemicals and hence determine the crop nutritional value and yield. This will affect directly or indirectly our diet and hence our health, since the micronutritional deficiencies or excesses of the products are often transferred to the consumers. Therefore, understanding the consequences of copper availability in the soils is a food quality and safety issue. Our European project, named TOMACOP, investigates the challenge through through a multidisciplinary approach, including molecular, chemical, physiological, and biochemical aspects. Results of these parts have been presented in other conferences, so today I'm going to focus on the effects of copper deficiency on the quality and nutritional value of the tomato fruit, showing sensory, nutritional, and post-harvest quality data. Under our Under our experimental design conditions, copper deficiency caused a reduction in the height and dry weight of the plant, as well as in the crop yield, as observed by the reduction in both the number of flowers and the number of fruits per plant. These plants were allowed to grow until fruit can be harvested, and 3 ripening stages were selected, focusing on green, orange, and red fruit. low copper availability did not affect the fruit color evolution, but reduced fruit firmness after green stage, as you can see in this first graph. Surprisingly, this fact was accompanied by a reduction of fruit maturity index as consequence of an increase in fruit acidity in orange and red ripening stages. That means that copper deficiency conditions provoke an overripe phenotypes in terms of firmness, but in turn reduce the internal maturity of the fruit. We also studied the micronutritional status of this fruit, and I show you here only the copper and iron data. And as you can see, the copper deficient growing conditions cause a decrease in copper content in all ripening stages. This difference was enhanced as ripening progressed, as indicated by the copper ratio between sufficiency and deficiency conditions. However, iron content increased in response to copper deficiency only in green fruit. We also analyzed typical compounds associated with fruit nutritional value. Total flavonoids and phenolic contents were barely affected by the growing conditions. Indeed, some differences in phenolics were found, but not a consistent pattern can be deduced from this data. However, total antioxidant capacity was constantly higher during the ripening and in the copper deficient fruit, in spite of lycopene content was reduced in orange and red ripening stages. After that, we evaluated the effects of copper deficiency on post-harvest fruit quality. And we found that the incidence of fruit cracking was increased by 7-fold in the copper deficient fruit, which makes this fruit completely unmarketable. It is important to note here that our experimental design ensured the same growing conditions for both set of plants, and hence we can eliminate the effect of other factors such as relative humidity, light, temperature on the incidence of these physiological disorders. So we can be quite sure that this difference in the incidence of fruit cracking was due to copper deficiency. In addition, we tested that the resistance to pathogen infection in this fruit, and we found that the susceptibility of copper deficient fruit to Botrytis cinerea infection was higher and faster as compared to the fruit harvested from the control growing conditions. In summary, agriculture is facing a challenge where copper deficiency limits crop survival, and the arable land often lacks this micronutrient. Copper-based fertilizers have historically overcome that challenge, but the governments demand that the overuse of these agrochemicals has to stop because of the environmental and human risks. Therefore, understanding the effects of suboptimal copper availability on crops' nutritional value and performance is needed to develop biotechnological strategies for future food safety and sustainability. Results presented here highlight that the deleterious effects of copper deficiency on tomato plant and fruit, and specifically on fruit quality and nutritional value and performance, which may ultimately affect human health. On the other hand, complementary results from our current project highlight the potential of copper transporters to be used as targets for biotechnological improvements focused on increasing crop resistance to copper deficient or toxic levels in the soil and also the use of these transgenic lines for phytoremediation. Last, I want to acknowledge the work of the students of our lab, as well as the collaboration of my colleague Maria Teresa Fuente. And also this work has been possible because of the collaboration with Dr. Lola Peña-Rubia from the University of Valencia and the Trace Elements group from the IATA in Spain. I want to thank also all the facilities used during the development of this work and the funding from the Marie Curie actions. And that's all. Thank you all for your attention. You can follow us on Twitter or following the project QR code and contact by email if you have further questions. Thank you.